In this video tutorial, I am going to discuss about classification of enzymes. In the previous video, we had seen that there are total 7 classes of enzymes are there. And to remember all these 7 classes, we have very nice mnemonic, that is OTHLIL, that is OTHLIL T. So by these mnemonics, you can remember all the 7 classes of enzyme. Let's once again look at the names of these 7 classes. So class number 1 is starts from the O, that is oxidoreductases. Class number 2 from the T is the transferases. Class number 3 is the hydrolases. Class number 4 that starts from the L is the Lyases. Class number 5 from the I is the Isomerases. Class number 6 starts from the L, that is Ligases. Remember, don't ever confuse between class number 4 and class number 6. Class number 4 is Lyases and class number 6 is the Ligases. As both starts from the L, so in this mnemonic we will not able to call which one is the first. So this thing we have to remember, right? And the class number 7, which is recently added, it was actually added in the August 2018. It was added in August 2018, and the name of this class is the Translocases. Translocases. Let's look at all these classes one by one. So first class, that is oxidoreductases. Now in this class, as the name suggests, all the enzymes which catalyzes the redox reactions are included in this oxidoreductases class. Now see, we generally write redox. Redox word is made up of two things. One is reduction and second one OX means oxidation. Now rather than mentioning reduction and oxidation separately, we prefer to use this word redox. Why? Because most of the time, actually all the time, reduction is always coupled with the oxidation and oxidation is always coupled with the reduction. There is no any possibility of individual reduction reaction. Whenever reduction reaction occurs, simultaneously oxidation reaction also occurs, and reverse is also true, right? So both are always coupled, and that's why we generally prefer this term, that is redox reaction. Now, the second important aspect is that there are so many different and confusing definitions of reduction and oxidation. But to simplify the thing, as far as biochemistry is concerned, remember, reduction is any reaction in which hydrogens are getting added, so plus sign. And oxidation is any reaction in which hydrogens are getting removed, so minus H. Okay, so this is the easiest way to identify whether particular substance is undergoing reduction or oxidation. Now along with this hydrogen movement, also remember oxygen's movement. If oxygen is being removed from the reaction, we call it as a reduction reaction, and if oxygen is being added in the reaction, we call it as a oxidation reaction. So this is simple trick by which you can identify whether particular substance is undergoing oxidation or reduction in a simple way. Okay, let's look at one generic example of this class of enzyme. So suppose we have two molecules A and B, and let's say A has two hydrogen atoms. So both of these react, and if this reaction is catalyzed by the oxidoreductase kind of enzyme, then what will happen? These hydrogens will be transferred to this B molecule, right? So ultimately, what will be there? A without any hydrogen atoms plus B, which now has these two hydrogen atoms. So A plus B H2. So as you can see, AH2 is removing its hydrogen ion, right? So this we can say that from the removal, we can say AH2 is undergoing oxidation reaction. Whereas what about B? B is getting converted to BH2. So here as you can see, H2 is being added in this reaction. So whenever hydrogen atoms are added, we call it as a reduction. So this is a reduction process or reduction reaction. Okay, so by this trick you can easily identify whether particular reaction is oxidation or reduction, and such kind of reactions are catalyzed by this oxidoreductases. Now let us look at one real life example of oxidoreductases class. So one enzyme is there which is alcohol dehydrogenase. Dehydrogenase. Now, what this enzyme does, it converts alcohol to acetaldehyde. So, let's take an example of alcohol. We have ethanol, and the structure of ethanol is the CH3, CH2 followed by OH group. So, this is the ethanol. Now, what happens in this reaction, which is catalyzed by the alcohol dehydrogenase? These two hydrogen atoms are removed. So, of course, we need a second molecule which can take up this H2, right? So, for that, we have NAD plus, which takes up these two hydrogen atoms and gets converted to NADH plus H plus. And so, the remaining molecule will be this CH3. So, CH3. Now, this carbon will be only a single carbon. So, carbon with its OH group. So, we can write it as a CHO, and this compound is called as a acetaldehyde. Acetaldehyde. So as you can see that ethanol, by removing its hydrogen ion, it is undergoing oxidation, whereas NAD plus, it is undergoing reduction. There are also other examples are also there. For example, we have various oxidases. For example, cytochrome C oxidase. We have various oxygenases also. Oxygenases. For example, cytochrome P450 enzyme. Then we have another example, tryptophan dioxygenase. Tryptophan dioxygenase. Remember, there is a very interesting difference between oxidases and oxygenases, and this difference I will cover it in the next video. Okay. Now let us look at the second example or second class of enzyme, that is transferase. So class number 2 is the transferases. So as the name suggests, this class of enzyme, they transfer one group from one substance to another substance. So we can say they are involved in the transfer of group, right? So let's first look at the generalized example. Suppose we have two molecules, molecule A and molecule B, and let's say A molecule has a R group, right? So when A and B react, and if this reaction is catalyzed by the transferase enzyme, then what will happen? This R group will be transferred to this B compound or B substrate, right? So now we will have only A, and this B will be attached with the R group, right? So such type of reaction where this group is being transferred to another substrate, we call it as those enzymes will be classified under transferase enzyme. Let's look at one real life example of this class, transferase. So for that, we have one very good enzyme, that is glucokinase. Now what this glucokinase does, it takes up a glucose, and another substrate is the adenosine triphosphate, short form is the ATP. What this enzyme will does, this ATP has a 3 phosphate. So out of 3 phosphate, 1 phosphate is being transferred to the glucose, and it occurs on the 6th carbon. Okay, so now we will have a glucose. As it is on the 6th carbon of the glucose, we call it as a glucose 6 phosphate. And after losing the phosphate, ATP is getting converted to adenosine diphosphate. So here we can see that phosphate group is being transferred. Other examples are transaminase, which transfer amino group in between various amino acids, and we have transmethylase, which transfer methyl group. Now let us look at the class 3 of enzyme, and it is hydrolases. Hydrolases. Now hydrolases, what this class of enzyme does. So any enzyme which breaks bond. So breaks bond by addition of water. So if any enzyme catalyze such reaction, they are classified under hydrolases. For example, suppose we have one molecule where A and B group are bonded with each other. So this is only a single compound in which A and B both are bonded with each other. Now what happens? This bond is getting broken down by the addition of water. So now water molecule will itself get distributed between A and B. Let's say A get hydrogen atom and B get OH group. So ultimately it will be AH plus BOH. So such types of reactions are catalyzed by the hydrolases group of enzyme. One real life example of this hydrolases is acetylcholine esterase. Acetylcholine esterase. Its short form, popular short form is ACH, capital E followed by ACE. Okay. Now, what this enzyme does? This enzyme acts on acetylcholine, and as by the rule, it has to add water. So, water molecule is added. Now, this enzyme acts on this both substrate, and it is separated into the acetyl. So, acetyl gets converted to acetate, and choline is separated. Right. So as we can see here, bond is broken. Other examples are all digestive enzyme. Our all digestive enzymes are of this hydrolases class. For example, we have pepsin, we have lipase, like that. Another example is the urease. It is also hydrolases class of enzyme. Now let us look at the class 4 of enzyme, that is lyases. Now this class of enzyme, they are similar to hydrolases, but there is no any requirement of the water. So we can say lyases, this also breaks bonds, but water is not required. Okay. So let's say, for example, we have one compound in which A and B both groups are bonded with each other. So this enzyme, this class of enzyme, what it does, it simply breaks down this bond without addition of water. So A plus B both gets separated. One real life example of this lysis class is the aldolase. So the example is aldolase. What this aldolase does is that it acts on fructose 1,6 bisphosphate. So this aldolase acts on fructose 1,6 bisphosphate, and it breaks down fructose 1,6 bisphosphate into glyceraldehyde 3-phosphate and dihydroxy acetone phosphate. This is one of the very important reactions of the glycolysis. It is actually this 6 carbon compound is getting converted into 2,3 carbon compound. Okay, so this is the splitting reaction which is carried out by the aldolase, which is of the class lyases. Another example of this class is fumarase and histidase. Now let us look at the class number 5, that is of isomerases. So just like any other class, which are their names are self-suggestive, this class's name is also a self-suggestive. So we know about the isomers, right? Isomers have a same molecular formula, but their molecules rearrangement is different. So what this class of enzyme does? They just does a molecular rearrangement. They convert one isomer into another isomers. So they can either create optical isomers or geometric isomers, right? So this class of enzyme, it causes interconversion of various isomers. So suppose we have this molecule A or substrate A. So when this class of enzyme works on A, what will happen? There will be molecular rearrangement in the substrate A. So I will mention its isomer as a dash. So this reaction is very simple. One real life example of isomerases is triose phosphate isomerase. So enzyme is triose phosphate isomerase. So triose phosphate isomerase, it acts on glyceraldehyde 3 phosphate and converts it into dihydroxy acetone phosphate. Both are geometric isomers of each other. So this reaction is reversible. Generally, isomerase reactions are going to be reversible. Other examples of isomerase class of enzyme is retinol isomerase, and phosphohexose isomerase. Phosphohexose isomerase. One thing I forgot to mention is that there is triose phosphate isomerase, and this reaction is also very important reaction of the glycolysis, just like this one. Okay. Now next we will look at the class 6 of enzyme. Let's write it over here on a new page. So class number 6 is the ligases. So as the name suggests, ligase means to bind. So what this class of enzyme does, it binds two substrate together. So they binds two substrate together, right? Generally, so there will be a bond formation, and there is a bond formation between carbon to oxygen, carbon to sulfur, and carbon to nitrogen, and they generally utilize ATP. So high energy bond is required. Okay, so let's say for example we have two substrate A and substrate B, and when ligase class of enzyme works on this both substrate, what will happen? There will be a bond formation between A and B, and simultaneously, as I told you, they utilize this ATP. So ATP gets hydrolyzed to ADP and PI. So energy is transferred in for making this bond. One real life example is the acetyl CoA carboxylase. Acetyl coenzyme A carboxylase. What this enzyme does, it acts on two substrate. First substrate is acetyl coenzyme A, and second substrate is the carbon dioxide. And it makes up a bond between acetyl coenzyme A and carbon dioxide, and so both binds with each other and forms malonyl coenzyme A. And simultaneously ATP gets hydrolyzed to ADP and inorganic phosphate. Right? Other examples of ligase class enzyme. So other examples are glutamine synthetase, and PRPP, that is phosphoribosyl pyrophosphate synthetase. Remember, this is synthetase, it is not synthase. So there is a very subtle difference between synthetase and synthase. See, over here in this synthetase, this T is there. So remember, whenever the this T is there in the name synthetase, right? That extra T is there. Understand that it is of a class of ligase, and so it is utilizing ATP or any high energy phosphate bond. Whereas synthase, as they don't have any extra T, right? So extra T is not there. So they don't utilize ATP. So such names are given, and these are actually very confusing names. So you must be clear whenever you write synthetase or synthase. So by this trick, the idea will be clear. So if there is extra T, then it will utilize ATP. If extra T is not there, it will not utilize ATP. One major difference, all synthetase falls under this class of enzyme, that is ligases. Whereas synthase, they are not included in the ligase class of enzyme because they don't utilize ATP, right? Whereas this class, it utilizes ATP. Now let us move to the seventh class of enzyme, which is recently discovered, and name of this class is Translocases. Translocases. This class was added in the August 2018. Now here also, as the name suggests, it changes the location of the substrate. So this is very unique thing. Till now we had seen about the chemical reaction, but translocases, what they does, they changes the location of the substrate. So in other words, or in proper scientific word, we can say that they facilitate the movement of ions or molecules across the membrane or their separation within the membrane. So let me write down this complete statement, that they facilitate the movement of ions across the membranes. Remember, it can be ions or molecules, or their separation within membrane, within membrane, right? So schematically we can say that, suppose this is the cell membrane or any other membrane, okay? So what will be there? So this class of enzyme, translocases, they will be located as ion channels, right? Or it may be a protein part also. And what will happen if this substrate E is there? Suppose, let's say this is the inside of the organelle or cell, and this is the outside. So what will happen? A molecule will be getting transferred from in to out, or it may be other way around also. Okay. So one real life example is the H plus transporting ATPase. So here what this enzyme will does is that in this cell, in this membrane, this enzyme is located over here. It will be transversing the cell membrane. Okay. So what will happen? H plus ions will be transported across the cell membrane. Okay. This is this H plus transporting ATPase. It is shown by this green barrel. So this action is carried out by the H plus transporting ATPase. We have one more example, is the ascorbate ferry reductase. Ascorbate ferry reductase. So this completes our discussion about the classification of enzyme. If you have any query or confusion, please write it down in the comment section below. Thank you.